basically, what is oral squamous cell carcinoma? So basically, it's a cancer or oral cavity cancer, and it's a prevalent cancer worldwide with a great amount of patients in India as well, with a high incidence and mortality rate, particularly in India. The lack of a reliable biomarker for early detection often leads to the late diagnosis and poor survival rates in the disease. So basically, what is OSCC for the reference? OSCC is generally a cancer which is caused by the excessive smoking, alcohol consumption, and to a greater extent, chewing or keeping tobacco for a longer period of time in the mouth. Now, how does this relate to my research article or the manuscript which I was being provided in the first assignment? So basically, the research article which I was being provided with in the first assignment was global proteomics based identification and validation of thymosin beta 4X linked as a prognostic marker for head and neck squamous cell carcinoma. Now uh, the article which I chose was regarding the same thing, in that it was given that in the article, marker for head and neck squamous cell carcinoma were researched upon, connecting it to the OSCC, as it is most common oral cancer in the squamous cells in the head and the neck region. This study aimed at the identification of potential biomarkers, salivary biomarkers to be precise, for OSCC using targeted proteomics. All right, so moving on to the study design and participants. In this study, the study involved 50 biopsy confirmed cases, that is, 50 confirmed patients with their biopsies done, which confirmed the results of OSCC cases, and 49 age and gender matched health controls as well. So 50 biopsies which were confirmed cases of the OSCC, and 49 others that were age and gender matched with the patients that had the cancer, and 49 others that were healthy individuals in the control group. So now the participants were recruited from the Department of Radiotherapy and Department of Otolaryngology at Postgraduate Institute of Medical Education and Research in Chandigarh, India. Now, the process was that unstimulated saliva samples were collected from each participant after a period of fasting. The study followed the remark criteria for reporting tumor mark prognostic studies. So basically, what is remark criteria? So remark criteria is nothing but reporting recommendation for tumor marker prognostic studies, remark. So it's an abbreviation used for the procedure that is being carried out for the reporting and recommendation of tumor marker, which are used in the prognosis and the study. In this case, 50 cases with biopsy were confirmed, and the controls were in the control group, 49 patients or healthy volunteers were taken, which had the same age and gender matched with the 50 cases that were confirmed. And the saliva collection was sorry, the saliva collection was unstimulated saliva samples, which were collected after a fasting period. Moving on to the next one. So protein selection. So basically, how was the protein selected that were to be identified in the proteomics? So basically, the candidate proteins were initially identified using a TMT tag-based relative quantification of salivary proteins in OSCC cases on LCMS. So what does this mean basically? TMT tag based relative quantification means TMT, that is tandem mass tag, which is a procedure where generally uh, the tumor research, that the tumor identification and the markers are being identified, in which the proteins are quantified using the TMT tag, and the salivary proteins in this case of OSCC, that is oral squamous cell carcinoma, were quantified on the basis of TMT tag, and then in liquid chromatography and mass spectrometry techniques were used in the protein selection. This analysis or the study identified 135 dysregulated salivary proteins. So from the affected individuals, 135 dysregulated salivary proteins were found in those samples, and the candidate proteins were selected on the basis of their gene ontology and protein-protein interaction network and the fold change of the protein, which was seen during the TMP tag quantification and LCMS studies. Uh, so basically, the dysregulated salivary protein were known to play a significant role in the cancer biology, were chosen for further analysis by PRM-based absolute quantification. So basically, what is this regulated salivary protein in the first place? So abnormalities in the protein translation and production, which lead to hyperproduction and ultimately cancer, ultimately adversaries or cancer in the cell. Now, 12 highly dysregulated proteins were identified in this selection process. Out of 135 dysregulated salivary proteins, 12 were highly dysregulated, and then after conducting the LCMS and other techniques of TMT tag identification, PRM-based absolute quantification of those protein was conducted. PRM, that is parallel reaction monitoring. This technique is based on high resolution and high precision mass spectroscopy. So as I mentioned, first of all, the researchers carried out TMT tag-based relative quantification, wherein they identified 135 highly dysregulated salivary proteins. In the second part, gene ontology and protein-protein interaction network analysis was done, and selection criteria for candidate protein was observed, and 12 highly dysregulated proteins were found in the third procedure, that is PRM, is parallel reaction monitoring based absolute quantification procedure. Now moving on to the next one. So significant protein dysregulation was seen in 12 highly dysregulated proteins. Out of those 12, 5 proteins were found to be significantly dysregulated in OSCC cases compared to the healthy controls. So basically, they studied OSCC samples and healthy individual samples, and they found that 5 proteins out of those 12 significantly dysregulated ones, majorly contributing to the OSCC cases. In the two protein, AHSG and KRT6C were significantly upregulated. So basically, these two proteins were significantly upregulated, while 4 proteins, that are AS, AZ, GP1, KLK1, BPIFB2, and LSCRT, were significantly downregulated. Out of these, LSCRT, that is the last one, was not detected in all the cases and controlled, and was excluded from the further analysis. So going on with in the next step, the researchers went ahead with AHSG and KRT6C as the significant upregulated ones, and AZGP1, KLK1, BPIFB2 as the downregulated ones. They found different levels, different amounts of the protein levels in the um, different levels of protein in the cases, the samples. I'm sorry, in the samples. So AHSG and KRT6C, those were the upregulated ones. So they discovered in the healthy individuals, the levels of these proteins were in the range of 0.65 for AS nanogram per ml of ASG and 0.93 nanogram per ml of KRT6C. Now, in the median level in the cases, so basically uh, the individuals that were affected for OSCC, they had 1.31 nanogram per ml of AHSG protein and 2.2 nanogram per liter of KRT6C protein. And the p-values are described as follows. So null hypothesis values are also mentioned here. In the downregulated ones, AZGP1 had median levels in controls group of 89.48 nanogram per ml, KLK1 2 we had 19.39, and DPIFB2 at 22.21 nanogram per ml respectively. And in the OSCC cases, AZGP1 was 16.87 nanogram per ml. KLK1 was found at to be 0.25 and BPIFP2 at 9.6. And they also had mentioned the p-values for these things as well. So we can say that significant upregulation and significant or drastic change in the downregulation was seen as well. Moving on to the next next one. So analysis of protein levels by the disease stage. So there are different stages in the progression of the OSCC in the tumor. So basically, the analysis of protein levels by the disease stage. There are various stages mentioned in the research paper, wherein the levels of 5 significant proteins were further analyzed sorry, analyzed and compared based on the disease stage. AHSG and KRT6C that were significantly upregulated into T3 and T4 stage, while BPIFP2 was significantly downregulated in T1 and T2 stage. AZGP1, mind again, the BPIFP2 was also part of the downregulated proteins. So basically, in T1 and T2 stage, BPIFP2 was seen as significantly downregulated, wherein AHSG and KRT6C were significantly upregulated. Their presence was seen in much more amount in T3 and T4 stage. Um, yeah, sorry, I'm sorry. So AZGP1 and KLK1 were significantly downregulated in both the cases of T1, T2, and T3, T4 stages. However, only AHSG and KRT6C showed a statistically significant difference between T1, T2, and T3, T4 stages. So in all the stages, T1, T2, and T3, T4, AHSG and KRT6C protein that are significantly upregulated showed a statistical significance of difference in all the stages. All right, so in T1, as I mentioned, in T1 and T2 stage, the BPIFB2 downregulated, BPIFB2 protein was significantly downregulated, and AZGP1 and KLK1 were also downregulated. Whereas in T3 and T4 stage, AHSG and KRT6C were upregulated, significant difference, and AZGP1 and KLK1 were downregulated. Moving on to the next one. Next one. So analysis of protein levels by regional lymph node metastasis. So basically T and M is a significant methodology in the uh, metastasis or the identification. So T basically refers to tumor, N basically refers to the lymph nodes. I'm sorry, N represents lymph node that has, or if it doesn't have cancer cells in the nearby or the vicinity lymph node. M refers to the metastasis. So in this one, the levels of 5 significant proteins were also analyzed based on regional lymph node metastasis. So basically referring that if at all the regional lymph node were affected by affected by the growth or metastasis of the tumor. AHSG and KRT6C that are upregulated proteins were significantly dysregulated in both NO and N plus stages. So as I mentioned, T and M method in the N, it refers to the presence or absence of uh, cancer cells in the lymph node or the nearby lymph nodes. So basically, NO refers to the condition wherein lymph node doesn't have or don't contain cancer cells. No cancer found in nearby lymph nodes. That is being represented by NO. And the other one is N plus stages. So N plus stages basically mean um, amount like the presence of cancer cells was detected in the lymph node region or surrounding lymph nodes, wherein wherein the BPIFP2 and KLK1 were significantly downregulated in NO stage only. So AHSG and KRT6C were significantly dysregulated in both the stages of NO and N+, wherein BPIFB2 and KLK1 were significantly downregulated in just NO stage only. So none of the proteins individually could be used to distinguish between NO and N plus stages. So their researchers say that there is no particular distinguishable difference or distinguishable characteristic of any proteins or presence of any protein, which can be considered for both the stages or individually as well. AHSG and KRT6 significantly downregulated in both the stages. BPIFP2 and KLK1 were significantly downregulated in just NO stage only. So coming on to the next part, that is logistic regression analysis. So the analysis revealed that AHSG, KRT6C, and AZGP1 together formed a risk prediction model for all the OSCC cases. So these three proteins were considered and were observed to be forming a major risk prediction model for the case for the carcinoma. And in this model had a statistical significant ROC curve with an AUC of 82.4%. The model was also found to be significant for late stage cases of the primary tumor, that is T3 and T4, with an AUC of 87.9%. So these are the ROC graphs that were presented in the research for the study. So as you can see here, the specificity versus sensitivity graph was made, and all the stages that are for the different stages of cancer in T1 and T2, these five proteins were uh, these five proteins were measured and plotted in the graph, wherein we can see different amount of significance of the data can be seen here. So the potential biomarker panels for OSCC diagnosis here. The study identified potential biomarker panels for the diagnosis of OSCC, that is oral squamous cell carcinoma cases, with the ability to differentiate between early and late stage of the cancer. The findings suggest that AHSG, KRT6C that are upregulated ones and AZGP1 together can serve, which is the downregulated protein, and so together can serve as potential biomarker panel for the diagnosis of oral cancer. Further validation in a larger patient population is needed to confirm these findings and explore the potential of these panels for clinical use. So the researchers researchers concluded that these three proteins, that is AHSG, KRT6C, and AZGP1, can be together considered for the potential biomarker panel for detection of or diagnosis of oral cancer in its early stages. Yeah. So here it is mentioned that how different panel can be used for the accuracy and uh, prediction of the prediction or early detection of the cases. AHSG, KRT6C, and AZGP1, as mentioned, can be potential biomarker panel for diagnosis of all OSCC cases. Potential biomarker panel for diagnosis of NO stage OSCC cases, that is wherein the no cancer found, the lymph node doesn't have cancer cells. In that case, the ALSD, KRT6C, BPIFP2, and AZGP1 protein can be used as a potential biomarker panel. And for N plus stages of OSCC cases, AZGP1 and KRT6C proteins can be used. So considering the potential biomarker in the proteomics, with the help of proteomics, OSCC, that is basically related to the head and neck region, carcinoma, um, could be